Okay, let's get started. Hello, everyone, and welcome to the ZOOMinar. Today, we are going to see the power of mass spectrometry to study protein aggregation in today's ZOOMinar. We have two great experts in the field as speakers, Dr. Brandon Ruotolo from the University of Michigan Chemistry Department, and Dr. Richard Vachet from Chemistry UMass Amherst. It's my pleasure to introduce my colleague, our first speaker, Dr. Ruotolo. He completed his PhD from Texas A&M and postdoctoral training at Cambridge in the lab of Professor Carol Robinson. His research is primarily focused on developing ion mobility mass spectrometry to study protein aggregation as well as the structural biology of membrane proteins. His excellent research contributions have already been recognized by several awards and honors. I'll mention a few of those, including the NSF Career Award, the Agilent Thought Leader Award, and the Young Investigator Award from the Protein Society. So with that short introduction, I would like to invite Dr. Ruotolo to present his talk. Brandon, all yours.
Thank you, Rams, for the introduction, and also thank you to all the other organizers for this symposium series. It's been really exciting to watch on YouTube and participate in, and I'm looking forward to the discussion that follows the presentation today. So I also want to apologize in advance. I'm getting over a non-COVID related cold, so if I sound a little bit raspy, that's the reason. So the plan today was to talk a bit about the protein misfolding work going on in my laboratory. But before we get there, I wanted to spend some time talking about the general motivation for our work in this area and why we think that mass spectrometry is a really exciting technology to apply to lots of different protein misfolding problems. So on this slide, I'm showing you a diagram from this now 10-year-old Nature review from Ulrich Hartl, where he, I think, elegantly makes the case that protein misfolding is a process that's mitigated and controlled and managed in many cases by a myriad of protein interactions, right? So if you just boil it down to the individual biomolecule that you care about, it can occupy, obviously, its native configuration and also a range of other states that eventually lead to amorphous aggregates and amyloid fibers, which are, in many cases, coupled to debilitating disease. But you can see that the transitions between these different states are mitigated by chaperones. And in a broader sense, the equilibria that protein undergo in the cell are all managed by actually a pretty wide network of different protein interactions and other biomolecular interactions that include cell membranes, metal ions, and other cofactors, along with the context of chaperones, roughly 180 different components or protein associated with that pathway. For the ubiquitin proteasome system for degradation, we're talking about 600 different components. And for autophagy, something in the neighborhood of 30 different components. So the big take-home message, I guess, from this slide, and one of the reasons that we think mass spectrometry is a powerful approach to this area, is that protein misfolding really takes place in the context of a complex mixture that's chocked to the brim with macromolecular protein complexes that one wants to interrogate structurally in terms of how they're interdicting on or mediating the protein misfolding process. So if you take that lesson and shift your gaze to therapeutic discovery, you start to think about, I think, the extreme polydispersity of the protein misfolding problem. If you just narrow your gaze on the targets themselves and forget the constellation of protein contexts that I just discussed with you, and say, for example, you focus on A-beta, which is a key molecule involved in Alzheimer's disease, or alpha-synuclein, which is a key molecule involved in Parkinson's. We'll talk about both of these molecules later on in my presentation. You begin to realize that even with these isolated biomolecules, you're dealing with a structural and oligomeric state level of complexity that is really hard to match throughout the rest of nature. Each one of these molecules can occupy a huge range of different conformations that are each individually coupled differently to different oligomeric states, dimers, trimers, tetramers that they can evolve into. And you really want to track all of this in the context of how they respond to environment. But also if you then build in just a single interaction partner, like a small molecule drug or a biologic, for example, then that already creates a different level of complexity because you have to worry about how each of those different conformational states, oligomeric states interact differently with the drug candidate of choice, right? So as you start to build in now this complex web, again, just to sort of really hammer home this idea of the complex mixture, if we have a single amyloid forming protein, like I'm showing you here on this diagram here in purple, and there's an interaction network of different regulation machinery around that protein, interaction partners, so on and so forth. And if you pile on top of that metal ions, small molecules, and a biological membrane to interact with, then you start to get a sense for the real complexity involved in protein misfolding diseases and what you want to track in order to understand how a therapeutic is really going to make a difference in the context of predicting its efficacy downstream. And so we feel if you're not taking complete hold of this complex mixture space, then it's easy to sort of fall into a space where you're making poor choices regarding drug discovery and development. And how to change this paradigm is really trying to develop technologies that can work with complex mixtures, but also extract structural information and get you details surrounding inhibitor binding and conformational transitions involved with that. And if done right, then this would also provide an equally opportune avenue to investigate the mechanisms involved in how small molecules and biologics in vitro and in vivo are interdicting with a protein misfolding target, and probably reveal a lot about the protein-protein interactions involved in how they regulate protein misfolding diseases. So to try and fill that gap, that technology space, my group has been pushing a combination of different Masterometry technologies. Some of these would be things that you may have heard of, and some maybe not. So we're going to spend some time at the beginning of the presentation walking through some of the basics. The first here is native mass spectrometry. So native Masterometry is a branch of Masterometry that is aimed at trying to take proteins in their native structural state, excuse me, and lift them directly into the gas phase for measurements. We try to do this while preserving, excuse me, sorry, non-covalent complexes between the protein target and therapeutics. Excuse me, sorry. One second. All right, let's try that again. So native Masterometry is a branch of Masterometry that's aimed at trying to lift proteins in their native structural state into the gas phase and directly measure proteins bound to ligands or other proteins. In fact, the technology can capture macromolecular protein complexes all the way up to the megadalton scale. Excuse me. And how this works is you take careful control over the ionization step. So you're trying to control the solution conditions of the proteins are exposed to just before ionization so that they're designed to mimic those that preserve native structure, as well as provide, excuse me, a route to lift those structures into the gas phase and preserve, technically trap those structures for analysis. So if we do our experiment the right way, and also make sure we don't energize the structures as they're leaving solution, we can trap structures that have strong resemblance to those that are present in solution. We get a range of charge states from the experiment. So different amount of charge is deposited on the surface of the biomolecules during the experiment. You can see in my cartoon here, I got a protein, excuse me, that has got a +3 and a +4 charge state. I can see if I do my native Masterometry experiment the right way, I can see ligand binding associated with a ligand attachment to the proteins. via mass shift because the ligand carries molecular weight. And from this experiment, I can not only track the structure if I do other types of experiments, which I'll describe in a second, I can also, by virtue of the relationship between the intensities of the ion currents that I can observe, excuse me, and their concentrations in solution, I can get KD values from the experiment, and as well as I can extract stoichiometry values in terms of the amount of ligands attached in different stoichiometries. I can do all of this from nanogram scale samples with no need to modify or label the samples. And I can do this in the context of complex mixtures. The other technology that my group deploys in our studies of protein misfolding diseases is ionizability separation. This is best thought of as a technology that's related to electrophoresis, which you're probably familiar with in either solution or in gel phase. In the gas phase, mobility separation is accomplished by injecting ions, a mixture of ions, into a chamber that has electric field superimposed across it. All these gray bars you're seeing in this chamber are electrodes that describe or used to anchor this electric field. Also in this chamber are these little yellow dots. They correspond to neutral gas. In our experiments, is a mixture of helium and nitrogen. So if I take these ions, expose the electric field, the field's going to drag them along this chamber. And as they get dragged along, they collide with the background gas. And the larger my molecule is, the slower it travels through this chamber in comparison to the more compact conformational states. So this is a size-based separation technology. It also depends upon charge, just like electrophoresis in solution or in gel. The difference here is that we can go a lot faster. We're on the millisecond timescale typically for these experiments. Again, that helps us connect the trap structures for their study. They're related to those in solution. There's also higher resolution, which we typically do in, at least in gel states. So we can separate size differences that are on the order of, in the experiments I'll show in native states, 2% different. For denatured structures and smaller molecules and peptides, you can actually resolve structures much, much more in detail than just 2%. And then you can extract from these drift times through this chamber something called the orientationally average solution cross-section, or CCS value. That is a measure of the orientationally average, effectively surface area of these biomolecules, which you can use as a restraint to build molecular models of the ions you're tracking in the experiment. So typically we put all these together in a single experiment. So we have the drift tube at the front of the experiment. We create our ions via native electrospray ionization, separate them by size and charge and mobility separation, and then sample the separated ions using time of flight Astrometry, which operates in the microsecond time scale, so it moves much faster than the ionability separation. But this allows us to track the mass, the size, and the relative intensity of each of the features in our experiments, separated along both those dimensional axes. And from this combined data set, which is usually plotted drift time on the y-axis, mass charge on the x-axis, we can capture small molecule binding, we can capture these KD values or binding affinities, we can track stoichiometries of binding, we can see chemical modifications that occur to the target, oxidations or other PTMs that might occur, conformational changes upon binding we can track, and we can also, again, build molecular models of all of this based on push and cross-section type restraints. So that type of experiment, where we're just tracking those types of parameters, is often enough to extract valuable information, but when it's not, my group has developed this idea of using what we call collision-induced unfolding, or CIU, to dig deeper into data sets and extract information surrounding biophysical stability from the ions as well. So in these experiments, what we do is we take a group of ions from a mixture. Let's say, in this case, we've got a population of ions that come from an antibody. And let's say we just care about 25 plus ions, so we can select out those ions to the quadrupole and measure the arrival time distribution of those ions possessed by ionability. We can check that that conforms to our expectations surrounding a native and compact structure. We can then carefully heat these ions, excuse me, over the course of the experiment by colliding them with background gas at larger and larger acceleration potentials. As we do that, the ion begins to adopt larger and larger structures indicative of unfolding. In my group tends to analyze the data in CIU fingerprint type modalities. Excuse me. Where we plot the drift time that the ions adopt along the y-axis and the collision voltage required to push the ion to these different conformational states along the x-axis. Sorry. So we've actually developed pieces of software that can go in and extract quantitative data from these types of fingerprints. We can identify features within them using a simple sort of first derivative type approach, and we can fit the features to logistic functions and use that to extract what we call CIU50 values, which represents the amount of voltage required to convert 50% of the preceding feature into its following feature. And larger CIU50 values, just like TM values, indicates an increase in stability for the structure. And you can track this as a function of ligand binding or different environments or modifications in sequence or mutation or what have you. Okay, so that's the primer on the technology we'll be talking about today. Let's go through a series of vignettes from my lab that illustrates the potential power of this technology for unraveling something about protein misfolding diseases. So the first little vignette I'll give you is about alpha-synuclein, which is of course a key molecule involved in Parkinson's disease. So PD is of course a debilitating illness, the whole range of really horrible symptoms that's affecting more than 10 million people worldwide at the moment. The key molecule here, as I've already mentioned, is alpha-synuclein. It's got a, from this NMR structure, you can see it's got this structured N-terminus and a relatively unstructured C-terminus. But this structure was determined in the presence of surfactant. And it's thought, and several other experiments confirm this, that the N-terminus of synuclein actually interacts with potentially cellular membranes to actually promote the order that you're seeing on this N-terminus. So the structural change is known, and it's known that biological membranes can be an important vehicle for trying to promote the structural change. Excuse me. What isn't as well understood is how biological membranes can influence the oligomeric states involved with synuclein. And so that's where we step in. So in our experiments, we're using lipid nanodiscs as a vehicle to understand how the synuclein is interacting with biological membranes. For those who are not interested, that don't know very much about lipid nanodiscs, these are effectively lipid bilayers that form discs circumscribed by belt protein. In our case, we're using a peptide called 4F to hold the disc together. They're about 10 nanometers in diameter, and they form like effectively a mini and very sort of tailorable bilayer structure for you to test various theories about how proteins and other biomolecules interact with the membrane. So here what we're going to be doing is exposing synuclein to these nanodiscs, comparing that to synuclein in solution. And in our experiments, what we'll probably be doing is lifting the complete synuclein nanodisc complexes into the gas phase and ejecting the synuclein off the disc to make measurements of the synuclein that was present in the disc but then released during the ionization process. We'll also be testing the impact of this compound called FNO75 on the synuclein. So FNO75 is a compound that my group, along with Matt Chapman's group, who really led the discovery on this, is working on as a generalized amyloid inhibitor. Matt's group actually discovered this first by looking at curly fibril formation and proteins involved in that process. But FNO75, before I leave that, it's a compound that's thought in other systems to stabilize larger oligomeric states, sort of a molecular chaperone, if you will, stabilizes the oligomeric states and prevents them to form fibers after that. So this is a snapshot of the raw data we get from the experiment. It looks complicated, and it definitely is, but I'm going to walk you through how we analyze this. So we go through all these different peaks that you see in drift time and mass charge space. And from that, we can identify signals that correspond to dimers and trimers and monomers, each of them in some cases bound to certain amounts of lipid. But all this is resolvable in the drift time Masterometry space. This group of signals up here that has this very sort of extreme slope in drift time versus mass charge space, these are all related to lipid clusters that are ejected off of the lipid nanodisk during the experiment. So we can remove all the chemical noise from the experiment and just focus on the mixture of different states that we observe for synuclein in the presence of these disks. And I'm just going to summarize the data here so we can track the oligomeric states from solution states, nuclein versus those in disks, and add or subtract FN075 here indicated as FN on this diagram. And you can see that we see a change in the amount of oligomeric state present for nuclein as we certainly add disks, and also we add FN075, especially in the dimeric states of the ion. We can also be using CIU. Again, larger CIU50s indicate more stability here. It seems like the disks in our experiments are increasing the stability of synuclein, which is maybe not super surprising. We can quantify that. For dimers, it seems like it stabilizes a bit more than monomers. So from these experiments, we can tell already that the membrane environment promotes alpha-synuclein oligomer formation, or likely it does, that membrane-associated alpha-synuclein oligomers are more stable than those in solution, and FN075 stabilizes membrane-associated synuclein oligomers to a greater extent than those in solution. So I'm going to switch gears now and talk about some work that we're doing jointly with Jim Bardwell's group. So Jim runs a group here at UM in MCDB. Jim's group discovered, or is really studying, this protein called small EDRK rich factor, or SURF, which they think is a modifier of a broad scale modifier of protein aggregation. So it's a highly conserved, intrinsically disordered protein. You can see the NMR ensemble structure here. So it sort of looks like broccoli, purple broccoli from this diagram. Only 4% of the structure is healable at 37 degrees Celsius. And it's known in C. elegans that SURF, or it's also called MOAG4, when you delete it from, or when you suppress it in terms of its expression, you see a lot fewer evidence of amyloid formation. So in this C. elegans experiments, what we're doing here is we are deleting the gene for MOAG4, and we see 75% fewer amyloid puncta associated with the poly-Q proteins linked to Huntington's disease. So it seems like the SURF protein is somehow accelerating or providing a template for more exaggerated or more complete amyloid formation or protein misfolding. So we want to understand with Jim how broad that property is. And so in Jim's lab, they did a series of THT type assays aimed at this type of question, where they looked at A beta 1 through 40, which again is linked to Alzheimer's disease, another amyloid forming or protein misfolding type target, where you can track how the protein aggregates by the incorporation of THT, which has got a selectivity for the beta rich sections of amyloid fibrils. So the more fluorescence you observe, the greater the fibril formation here. And you can see that, you know, A beta 1 through 40 definitely goes on to form fibrils, as you probably know. And this is the EM data that you can also see for the morphology of the fibrils. When you add SURF in fractional quantities relative to the A beta 1 through 40, you can see that it ends up accelerating the fibril formation mostly by truncating the lag phase associated with the fibril formation. And the structure of the fibers is somewhat changed, but somewhat similar as well. So it seems like it modifies and accelerates amyloid formation in vitro experiments. So we wanted to deploy ability Masterometry to really dig into the details here and figure out what's happening at the molecular level that drives this fibril formation acceleration, which seems to be a general feature of SURF when it comes in contact with other proteins. So this is the native ionizability Masterometry data set for surf, where we've added surf and A beta 1 through 40 in a 1-to-1 ratio. We can see signals associated with the surf protein from yeast. We can see signals for unbound A beta 1 through 40, and we can see clear signals here for the complex between surf and A beta 1 through 40 in a 1-to-1 stoichiometry. We can also observe other complexes at lower levels that include surf dimers, 2-to-1 and 1-to-2 complexes between surf and A beta also are detectable. And we probed samples that were extracted from every time point associated with the tracking of the fiber formation process associated with A beta 1 through 40 here. And these complexes that we're observing and showing to you here are only extracted from the lag phase of amyloid formation. So these occur early on, and afterwards we can't detect them. So clearly they're there in the early stages of viral formation, but after that, they rapidly aggregate and fall out of solution in a way that we can't detect by ionizability Masterometry. We also track the structures or the sizes of these different complexes. Now, this is the same data I showed you in the previous slide. We're just looking at the drift time resolved form of that data. We can track the same complexes, all color-coded here with these squares, so we can walk through all the different complex structures. Again, so just paying attention to the drift time dimension for this data, you can see a number of features in this data, especially some here related to surf, but also for some of the complexes here, exhibit very wide and broad arrival distributions in ionizability space, indicating a lot of structural polydispersity for these structures, which is not normal, right? So if you're typically observing a complex between two proteins, in our experience, what you might expect to see is a much more narrow distribution between the two, if it's a selective and pervasive and structurally tight complex. And then we're seeing sort of the opposite here, where if you see complex between surf and A beta, it seems like it at least retains that polydispersity that was present in the original proteins, but in many cases seems to exceed that. So yeah, these long arrival time distributions simply are a marker of how polydispersed structurally these complexes are. And if we convert the ionizability dimension into cross-section space with the same dimension that we would use for modeling some of these structures, you can get a better sense for how much structural polydispersity there is in the complexes here versus the protomers that give rise to those assemblies. So we have the size distribution here for surf and A beta, and the complexes are all up here at largely per unit charge, much higher cross-sectional areas than the individual proteins involved. And indeed, they're higher cross-sections than the arithmetic sum of the cross-sections involved in the complex in the proteins that gave rise to their structures. So these are, very large structures indeed. We can do the same experiment for alpha-synuclein with surf, and we can see again a range of complexes that form, identify all of the peaks in our spectra. We can see examples of 1-to-1, 2-to-1, 1-to-2, and 2-to-2 type complexes between synuclein and surf. We can track all of the structures for these ions, and again, the same theme pervades. Whereas if you compare surf and synuclein, which are indeed polydispersed in their own right, the complexes that they form are even larger and more polydispersed structurally than the proteins that gave rise to the complexes. You can see that in 1-to-1 and 1-to-2 complexes, as well as 2-to-1 and 2-to-2 complexes. And indeed, here's some oligomers of circ and synuclein for you to compare against here. And in many cases, these are very large, very polydispersed complexes, not what you expect for a specific binding interaction. And this plot is just designed to give you a sense, not just for the range of cross sections here, but also their weight from the point of view of our measurements. It's a violin plot. And you can see here, I think pretty clearly in the A beta 1 through 40 here, if we have A beta on its own, you bind it to surf, the polydispersity of the surf complex is much, much greater and much larger, there's a larger surface area probably, than the individual proteins involved, indicating a potential route for how this must form an acceleration platform for further fibril formation. And the same is true and simply more exaggerated of larger cross-section because of the larger sense of synuclein for these types of complexes too. And these data really form the very first observations of direct interactions between these two different classes of proteins. So from this, we constructed a simple map, if you will, a diagram that sort of tries to illustrate what we think is happening mechanistically here. And really, it's about SERF interacting with the more unstructured forms of A beta or synuclein and preserving those unstructured forms in the elongated state that then is pre-organized then for accelerated fibril formation. And this was published in PNAS a couple of years ago. All right, so the last vignette I want to talk to you about is our more recent work associated with biotherapeutics aimed at A beta and Alzheimer's disease. So this is a controversial area at the moment, as many of you know. So aducanumab, the very first biologic approved by the FDA just last month, has suffered a lot of controversy during its clinical trial phase, lots of disagreement surrounding how effective this must be. We wanted to step into this space to ask questions about the details of how this type of biologic, indeed other biologics in the space, interact with A beta, which proteoforms interacts with. And we feel it's important because the approval here, regardless of what you think about it, is certainly going to set up a situation whereby a greater number of these types of A beta targeting biotherapeutics are going to come on to the clinical trial space and past. And so developing greater degrees of chemical measurement prowess that are aimed at trying to understand the details of how these biologics work is going to be valuable for trying to assess their potential efficacy down the road. Right, so really we're going to be trying to target on basically how how we can sort of move beyond typical clinical assays that are aimed at trying to evaluate how A beta interacts with these types of biotherapeutics. So, a snapshot of how this typically works is usually using native gels. And this is an example of data from crenezumab, which is a Genentech's now defunct antibody in this space. It gives you a sense for, the level of information content you can extract from these types of data. And we're trying to do much better than these types of measurements and give you a lot more detail surrounding the individual stoichiometries and specificities of the A beta, and maybe something structurally as well about how the complex is formed and to what extent they can be stabilized. All right, so this is work that we're doing with Pete Tessier's lab. So Pete's a professor here at UM in chemical engineering and also the Biointerfaces Institute. So we're taking 3 different antibody-based therapeutics that are aimed at A beta. Aducanumab, the famous one from Biogen, crenezumab, the Genentech antibody, and A34, which is a version of aducanumab that Pete's group has developed that is meant to be an improved version of aducanumab in terms of how it targets A beta. We're going to look at 3 different A beta proteoforms here, 1 through 40, 1 through 42, and 3 through 40, that are all biologically relevant forms of A beta. We're also designed to test something about how the interplay between epitope sequence and conformational epitope might regulate the interactions between the A beta and the antibodies here. We have 2 different concentrations to pump up the oligomer population A beta, 30 micromolar, 100 micromolar, and we're all going to pre-aggregate the A beta before we do the experiment. We're going to measure emissibility Masterometry data and CIU. So we can see just from the original sort of stock data, this is without any A beta added, we can see good looking data for all 3 antibodies. We see some strange looking satellite peaks here for A34, but this is, remember, academically produced antibodies, so there's going to be some level of impurity here. We think this is just a small sequence addition to the classical antibody type they're aiming for here, and doesn't really affect our ability to measure the effectiveness of A34 in the experiments. We add A beta at 30 micromolar to these samples, and we can see a range of complexes form with the antibody. And we can see this via mass addition, simple native Masterometry. We can see 1-to-1 and 1-to-2 complexes for both aducanumab and A34. And we can see exclusively 1-to-2 complexes with crenezumab. We do the experiment with 1 through 42, and we see essentially the same sort of thing we're seeing with 1 through 40. I'll show you the 3 through 40 data where we're picking out the little N-terminal sequence there at the beginning of A-beta. This is the actual epitope that aducanumab targets within A-beta, that N-terminal region here. So as expected, we don't see any binding for aducanumab or A34 really to A-beta 3 through 40, but we do see binding to crenezumab as expected because it's targeting a different epitope. I also want to make the point that we do see plenty of free oligomers of A-beta in our samples, even when we're doing the binding experiment, because we're loading them up. And we can also measure binding constants for these. And in general, we measure crenezumab as having a much greater binding constant, a much lower KD than either aducanumab or A34. But just to give you a survey of all the complexes that we're able to track in our experiments, so at low concentrations of any of our samples, we see up to 1-to-2 complexes. At higher concentrations, we can see up to tetramer binding, which indicates that there's some efficiency and some effectiveness for binding larger oligomeric species, probably, for especially A34, but not so much for aducanumab. And crenezumab seems to also have the ability to bind these smaller oligomers. We can also track the stability increases that one adopts when you form these complexes using CIU. And we can track that for both the first transition from this ground state feature to this first and polar feature, as well as the second transition from this middle feature to this final feature out here. That's what these two different bars here are for each of these data sets. You can see that, as you might expect, as you bind the antigen to the antibody, you get an increase in stability for the overall complex. But that slope of increase in stability is different for each of these antibodies. Indeed, A34 shows the strongest slope. Aducanumab shows a steeper slope still, and crenezumab shows a much shallower slope than both of those two. Okay, so I want to stop there and transition to discussion. I hope I've given you a bit of a survey here in terms of how powerful ability Masterometry operated in this data mode can be for trying to untangle some of the long-standing problems and mysteries in protein misfolding diseases. We think our nuclein data provides some unique insights into how biological membrane can promote oligomer formation in that context. Our SURF data, I think, is also really exciting because it creates a really first-in-class data set that shows mechanistically how that protein is templating accelerated fibril formation. And we're especially excited in our aducanumab and biotherapeutic data because we think it's going to pave the way into the future, sort of giving us a really, I think, powerful tool for assessing the details of how these types of antibodies can be developed and discovered in a way that really digs down to a level of detail that allows you to make really good choices about which of these antibodies might be really good to push forward in terms of discovery and development cycles. So I thank the people that did the work here. So Varun Bidarki, a postdoc in my laboratory, responsible for the work I showed you with Jim Bardwell's lab. Yilin Han is in charge of both that nuclein data I showed you, as well as the aducanumab, the biotherapeutic data. Collaborators, Matt Chapman, Jim Bardwell, Pete Tessier. I want to thank our sources of funding, mostly from the National Science Foundation for these projects. And again, thank you for the invitation and for your attention. I'd be happy to answer questions.
Thank you very much, Brandon. Great talk. Are you able to see the Q&A folder at the bottom of your Zoom link or screen?
Okay.
I can read the questions for you going from the top. There's a question from Sikan Liu. Thank you so much for this wonderful talk. I was wondering whether you have tried SURF with the A-beta 42 apart from A-beta 40.
It's a great question. Yes, we have. It, I think, behaves very similar to A-beta 1 through 40. It's just hard to work with, so I didn't show you the data, but we have that data and it's part of that paper.
The second question from an anonymous attendee. How much heterogeneity in the data is expected when there is a stable one-to-one complex? In other words, do you have examples where there is no heterogeneity in the mass spec data?
Yeah, that's a great question. So I think that's the norm, especially when you have soluble proteins that come together to form complexes. You would expect to see a very narrow and tight distribution in drift time that's near the resolution limits of the instrument, forms a sort of Gaussian profile. It's rare to see the level of polydispersity I showed you in that CSURF data, which is why we're excited about it, because I think, if you look throughout the literature where people have used this technology to look at protein complexes, 99% of those complexes look like, tight, structurally sort of almost rigid complexes that form, and you can see that in ability distribution, but this is not the case in those misfolding data.
The next question is from Christian Grisinger. Why do you find monomer binding to aducanumab? It is supposed not to bind the monomer, or do you have an oligomer of A beta, say 10-mer, to which then 10 to 5 compounds bind?
Yeah, so so it's interesting, right? So I think that our data definitely shows that it does bind monomer to some level of efficiency. We're still working out, so we see both 1-to-1 and 1-to-2 stoichiometries. It's hard to know if that 1-to-2 is simply both elements of the Fab binding individual monomers, or that's a dimer-related binding efficiency. We're going to be using HT exchange to help figure out, which of those situations is reality. But yeah, the only so it's in stark contrast, I think, to the crenezumab data, which only shows 1-to-2 stoichiometries with no evidence whatsoever of 1-to-1 binding. There, we don't know if it binds monomer or not. It definitely has a tighter binder of A beta than aducanumab in our experience, and it's possible that it's binding dimer exclusively, but it's also potentially likely that it binds monomers with really great efficiency, so it soaks up 1-to-1 complexes on both the Fab arms very, 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 very readily, and that's where you get the 1-to-2 stoichiometry. So we're still trying to work that out. So the last part of the question, you have an oligomer of A beta, say 10, to which you then 10 to 5 aducanumab 5. Yeah, I'm not sure I understand that part of the question. We definitely tried to.
Many compounds binding to oligomer.
Yeah, so we don't see any evidence of that, like evidence of one or a few A beta nucleating lots of antibodies. Like I said, we definitely tried to produce a situation where there are plenty of A beta oligomers around, up to, I think, tetramer A beta in our experiments. And like I said, only when those concentrations were made pretty substantial did we see any evidence of oligomer binding for any of the antibodies. But again, this is like a very beginning. I think we collected this data a few months ago. I think we're excited about where it can take us in terms of understanding the mechanisms that underlie how these antibodies actually work to bind A beta. It was shocking to us how little was actually known about the selectivity for some of these antibodies with respect to these different proteoforms and structural states of A beta. And I think this technology could really be valuable for unpicking that.
Just for the sake of completeness, could you comment on the does this binding with the compounds, does it vary with salt, buffer, pH, other conditions? And how do you prepare your oligomers?
Yeah, so that's a good question. So we prepare the A beta oligomers in a way that's very similar to how we prepare them for analyzing interactions with small molecules. So we prepare them at slightly basic pH using incubation periods designed to pre-nucleate some sort of oligomer population. Then we spin off any of the formed like insoluble oligomer and then just look at the soluble fraction from those experiments, designed to basically sort of like try to arrest a population of oligomers that are present in solution that would be like prone to form fibrils, but we're trying to slow the process so we can capture them and make measurements. We haven't probed how like different preps, other than that concentration ramp I showed you between 30 and 100 micromolar, how that influences binding. I think it'd be interesting to look at like pH, for example, or maybe formulation in the context of how you prepare the antibody, like different salts, for example. That would be interesting to look at, and that's part of our future plans.
Great. Next question is from Ashutosh Tiwari. For a protein ligand protein interaction, what is the range of KD values that can be measured by mass spec?
Well, that's a great question. So I would say we can certainly go as tight as nanomolar in terms of measuring a high confidence KD value. Going too much tighter than that is problematic because, again, you're limited by the detection limit of the technology. So you want to be able to observe both apo and bound state in order to get the ratios of both. And so we're limited at the bottom level by the limit of detection of the instrument. And we simply can't measure below nanomolar amounts of the biomolecule. So we can't go to lower concentrations than that to probe like very tight binding. For weaker binding, I would say we've gone up in my lab to millimolar type KDs. There you have to be careful because if you pack lots of biomolecule into ionization source, you can easily form artifactual complexes that are product not of binding in solution, but of drying in electrospray. But if you can carefully control for that by taking compounds you know don't have interactions with your biomolecule and looking for when you see those artificial complexes form and then normalizing that out of your data. And if you do that, you can get up to, I would say, millimolar type KDs. But that's really the range. I would say nanomolar to millimolar, probably.
So Varun has responded in the chat box. SURF was 10 micromolar by mass spec. That's what he says. Okay, so the next question from Ashutosh is, what is the normal role of SURF, and is it concentration? Does it concentration change in the disease state?
What is the normal role of SURF and I don't think its normal role is completely known or understood. Like I said, it was discovered as this sequence that if you deleted it, it seemed to improve the amyloidogenicity of certain targets within biological systems. But I think beyond that, I don't think it's completely known what it does. What's the other side of that question?
Does its concentration change in the disease state?
That I don't know either. I'm guessing it probably does, but I don't know.
Okay, let's keep the remaining two questions toward the end of the ZOOMinar in the interest of time. So let's move on to the next speaker. Thank you very much, Brandon. So you can introduce Richard. Richard, you can share your screen.
Okay.
Yep.
All right. Excellent.
Okay. So it's my pleasure to introduce Richard Vachet, who's from the University of Massachusetts Amherst and a longtime friend and colleague of mine. So Richard got his training, his Bachelor of Science in Chemistry from College of William and Mary, and did his PhD in analytical chemistry at UNC Chapel Hill with Gary Glish. After that, he did a postdoc at the Naval Research Laboratory with John Callahan, and moved in to be an assistant professor of chemistry at University of Massachusetts Amherst in the late 1990s, and rose to the ranks, and was just recently head of the chemistry department at UMass Amherst, and I guess gratefully gave up that position in 2019. Richard's group, I think, is very well known for developing chemical labeling strategies combined with Masterometry to ascertain the structures of a variety of proteins, especially those involved in protein misfolding diseases. He's won a number of honors and awards for this, too many to mention quickly here, but in 2019, he won an Outstanding Research Award from UMass Amherst. In 2011, he was a Distinguished Teaching Award finalist, indicating how well he presents his material. I'm looking forward to a good talk there. In 2004, he was a GlaxoSmithKline lecturer in analytical chemistry at UNC Chapel Hill. With no further ado, looking forward to your talk, Richard.
Thank you, Brandon. Thanks for the nice introduction, and thank you to all of you for joining us on this Saturday to hear about what we have to say. Thanks, Rams, for the invitation too. It's nice to be part of this group. So I'm going to tell you today just some little tidbits about some work that's been done in my group over the past several years, in which we really have been motivated by developing some new mass spectrometry related tools to address problems in protein chemistry with a particular focus on protein amyloid formation. And as the title of the talk suggests, some of the early stages of protein amyloid formation. And so I thought I would start off by telling you a little bit about the the test bed system, or the amyloid forming protein that we have been most interested in over the years, and it is Beta2 Microglobulin. Many of you probably are familiar with this protein. It's one of the more well-studied amyloid forming proteins. If you're not familiar with it, it is a relatively small protein, 99 amino acids. It's a subunit of the class 1 major histocompatibility complex. And it's normally a fairly uninteresting protein. And because in most of us, it's on all of our nucleated cells. We have a fair amount of it in our blood at any given time. But for those who undergo long-term hemodialysis because of kidney disease, this protein can form amyloid deposits, amyloid fibrils in the joints. And so all patients on dialysis for a long enough period of time will develop these amyloids. And there's currently this condition, dialysis-related amyloidosis, or DRA. There's no treatment for it beyond getting a kidney transplant. But for those who are unable to get that for one reason or another, this is a pressing condition and a challenging one for them. Clearly, kidney disease is bad enough and dialysis is bad enough, but this adds to the problems. Now, just a little bit about what's thought to occur with the amyloid formation of Beta2M in vivo. The precise mechanism isn't worked out, but what we do know is that the protein, like I mentioned, is a subunit of an MHC complex in all nucleated cells. As normal cell turnover occurs, the protein gets shed, it ends up in the bloodstream, and in most of us, I'm pretty sure here, the concentration is somewhere in the 100 nanomolar range in our blood at any given time, and it gets degraded well by the kidney. In fact, it's one of the probably 40 or 50 most abundant proteins in our blood at any given time. Those, like I mentioned, who have renal failure, this protein builds up in the serum up to 50 or 60-fold higher than normal. And then via conditions that are not exactly known, the protein can aggregate and ultimately deposit as amyloid fibrils, as I mentioned. Now, the high concentration alone is not sufficient. It's necessary, but not sufficient. There are a couple other conditions in which Beta2M will have a higher concentration and not form amyloid. So there's something unique about dialysis that causes it to occur in addition to the high concentration. What that is not known, but there have been numerous ways that have been shown in vitro to simulate the amyloid formation of Beta2M to form fibrils that are like those that are extracted from patients. Over the years, people have studied things like low pH. A lot of proteins will form amyloids at low pH. At more physiological conditions, there's been other factors such as the presence of stoichiometric amounts of copper, or a mutant of Beta2 microglobulin called delta N6, in which the first 6 amino acids have been cleaved, and in a prion-like fashion, this protein can convert Beta2M to amyloids under the right conditions. Other factors like trifluoroethanol or collagen or reducing agents have also been shown to be able to in vitro convert Beta2M to amyloids. Now, like a lot of amyloid forming proteins, the process involves some partial unfolding, some oligomerization typically, and then ultimately formation of the fibrils. And what my group has mostly been interested in has been these early stages of this process and trying to develop tools, like I mentioned, that really allow us to tease out some of the molecular level details of what goes on in these steps. And really, one of the motivating factors really been twofold, two aspects that have motivated us. One has been the ability to develop some tools that will provide structural insight in cases where other more traditional biochemical and biophysical tools are difficult or impossible. So that's been one motivator, and obviously mass spectrometry has been one of the key tools we've used. But we've also been very interested in leveraging the information that we learn to study ways or develop and find out ways to inhibit the process. And at the end of my talk, if I have time, I'll tell you a little bit about the work in that regard that we've done. So right now, or at least for the story that I'm going to tell you today, most of what I'm going to tell you about is that we have been very interested in understanding how copper can initiate beta-2 amyloid formation. My group and Andrew Marenker's group at Yale University, in fact, Andrew's group was the first to discover that stoichiometric amounts of copper can convert wild-type beta-2M from being monomeric to being amyloid fibrils. Over the years, we have studied some of this process and tried to sort out some of the oligomeric changes that the protein undergoes as it goes from monomeric to fibril. And what we found is that upon binding copper, we get the production of a series of oligomers, discrete oligomers, that are often present pretty quickly after adding copper. We have dimers, tetramers, hexamers, and then typically not many things larger than that before we get to amyloids. And so there's the uniqueness of this process has been intriguing to us over the years, because we see these oligomers that go as pairs, dimers, tetramers, hexamers. We don't see trimers, we don't see pentamers. And so that it indicates a degree of organization that's important in this process. One of the things that's been particularly interesting for me, although I'm not going to talk a lot about it today, is the fact that copper has sort of a catalytic role in this process, where it's necessary to get the process going. It's necessary to assemble the oligomers, but then copper is lost, and that there are actually copper-free oligomers that are formed, and the final fibrils themselves have no copper associated with them. And so this catalytic process, we've actually been able to look at even at substoichiometric amounts of copper and can convert it to amyloid at relatively low concentrations of copper, which is interesting in its own right. But what I'm going to do today is tell you about some of the tools that we've developed and we've used to really shed light on what some of the structures of these pre-amyloid forms are, what are some of the structural changes that the protein undergoes upon binding copper. And in particular, we've used native mass spectrometry, which Brandon just talked about. He also alluded to in my introduction that we have been using some chemical labeling tools to study proteins, and I'm going to spend a good amount of time talking about that, but also ion mobility and computational modeling, which really is helpful for taking what ultimately is low resolution structural information and adding some more detail to it. So mass spectrometry is not the first thing you're going to think about necessarily if you want to get structural information about protein oligomers. You know, certainly it can tell you stoichiometries, you can tell you if you have a dimer or a tetramer. But I have a thought experiment for you. This is usually easier to do when I'm in person, but I'll do it anyway. You know, so the question is, if you had a series of dimers that could be arranged in all sorts of fashions, how do the masses of these following shapes differ? Now, that's a trick question, of course, because they don't. This is the nature of isomers or conformational isomers. They don't differ in mass. And so in order to get some insight into the higher order structure of oligomers or to proteins, there's a number of things we can do. And one of the things that we like to do is we try to do some chemistry on the proteins in solution that allow us to encode structural information into the protein's mass. And then we can use some of the wonderful tools that mass spectrometrists and people that are involved in proteomics have been using for years to sort out where that chemistry has happened and deduce structural information for that. Now, when we do this in mass spectrometry, there's several different approaches that people have used. Hydrogeneterium exchange is one very common one, in which the hydrogens that are normally associated with exchangeable sites, particularly those on backbones when we do mass spectrometry, can be exchanged for deuterium, and the ones that are exposed to solvent or easily or not tied up in hydrogen bonding exchange faster than those that are buried or involved in hydrogen bonding. And you can do that over time and then use mass spectrometry to read out which ones have undergone exchange in deuterium and which ones have remained hydrogen. and then you can deduce some structural information that way. Another approach is cross-linking, where you basically rely on trying to identify those amino acids that are nearby in three-dimensional space, even though they may be far from one another in primary sequence, and chemical reagents are used to do that. And you chew up the protein to go find those sites that are near one another. And then there's another technique that I'm going to really focus today on that I refer to as covalent labeling. Others have referred to it as protein footprinting, where the idea is we take a protein, react it with some reagent. Unlike cross-linking, it is a monofunctional reagent. And so what happens is that we react with solvent-exposed amino acids on the surface of the protein, and we sort of paint the protein in a way. And so that's what I'm going to spend most of my time talking about, as I mentioned. Now, the way to think about how this could be used for studying protein structure is can be shown in this little image here, where it's often done in a comparative way, where we compare maybe a monomeric form of a protein to, say, a dimeric form of the protein. And the idea is that this reagent that can react with solvent-exposed amino acid side chains, we can do the chemistry in the presence of the monomer or the dimer, and then in the presence of the dimer, those sites that are at the interfaces of the protein now will no longer be reactive because they're buried away from solvent and not accessible any longer to the label. Because it's a covalent reaction, we can use the wonderful tools available to us from proteomic studies to do enzymatic digestion, and then LC-MS and MSM analysis to find and pinpoint the particular residues that have reacted. Now, there's a number of different reagents that people have developed and used for this purpose. Our personal favorite is diethyl pyrocarbonate, which structure is shown here. It's a very electrophilic compound. It's been around and well known for a long time. People have used it for years to inactivate proteins who have histidines in active sites because it reacts nicely with histidines. But it turns out it reacts with a number of different, in fact, all of the nucleophilic side chain ones, are at least reasonably nucleophilic side chains in proteins, histidines, tyrosines, lysines, threonines, and so on. We like this reagent for a couple of reasons. One, it's incredibly simple to use. It's as simple as adding it to solution, and it reacts very rapidly. It also provides products, a single product that can react with these different residues, and it can account for about 30% of the, or at least cover 30% of the average protein's structure. So it provides good surface coating, if you will. So what I'm going to tell you about is a little bit of the work we've done to try to use this reagent and some other tools to identify and get insight into the structures of the pre-amyloid oligomers of beta-2 microglobulin. Now, one of the nice features of this chemistry, and I guess mass spectrometry in general, or this chemistry in general, is that it can be done very fast. And so, for example, if we do this experiment with beta-2 microglobulin under amyloid forming conditions, like I mentioned, we're very interested in copper, converting it to amyloids. You can add copper, and that acts as a starting point for the amyloid reaction. And over time, in solution, pretty immediately, you get some dimer. Eventually, the dimers go to tetramers, and tetramers to hexamers, and so on. But at pretty much all the time points after adding copper, we almost always have a mixture of species, monomer and dimer, monomer, dimer, and tetramer. And so when we do this covalent labeling reactions, the idea is that we start the reaction, and then we'll take aliquots, do 1-minute reactions with DPC, then digest the protein, do LC-MS, and then repeat this over time as dimer increases in solution. When we do something like this, we can then map and measure the labeling extents for many different residues on the protein, like I mentioned, about 30% of the protein surface. And so we can, for example, and pinpoint this to specific amino acids. And so by and large, over time, as we do this chemistry and we look at the level of modification, we'll see things typically fall in one of two categories. One category is we have residues like lysine 41 here, which largely remains unchanged over time within error of the measurement, or other ones like histidine 13 that decrease in labeling over time, suggestive of its reactivity or it's being buried in some way as the dimer is formed. Remember, it's not going quite to zero. It may eventually, because we have a fair amount of monomer that effectively act as a, where there's monomer that has histidine, presumably that's exposed, and then the dimer that maybe has a cysteine 13 that's buried. Now we can take all of this labeling data for up to 25 residues in this case, and if we take this data and just simply map the results onto the known crystal structure of the monomeric form that there are many of, there's NMR structures as well. And what I'm showing here is a picture of Beta2 Microglobulin, which is, by the way, a beta sandwich fold, where if you want to look at it on your hands, it's a 4-fingered strand and a 3-fingered part of the bread of the sandwich. And if we map this, where in blue we show the residues that have decreased in reactivity, and in red we have ones that are unchanged, sort of like lysine 41, or maybe have slightly increased, which a couple of residues do, you can see pretty quickly that all of the decreases happen on one face of the protein. In fact, on the 4-fingered strand, if you will, or a 4-fingered sheet. And so fairly immediately, you get some insight into the fact that perhaps the dimer is forming by these two sandwiches, or 2 4-fingered strands coming together to make a dimer. Now, that's admittedly fairly low resolution because we're only mapping 25 residues at a time, and we have the benefit of just mapping that on the known structure. But we want to take this a little bit further to try to really understand more deeply and develop models of what this dimer could look like using that covalent labeling data as some constraints. And so what we did is we did some modeling. We again began with a monomer. We modeled in the copper binding site, which we had experimentally determined many, many years ago. And then after modeling that in, we docked the copper bound monomers together and generated thousands of structures. And then we did MD simulations of these structures for up to 1 microsecond of simulation time, and looked to see if there were structures that were stable for that long. And we found that there were 3 stable families of configurations. And those families were largely, and we did both with and without copper, and we found that 2 of the important ones were what we call a head-to-head interaction and a side-to-side or side-by-side interaction. And long story short, the side-to-side configuration is almost perfectly consistent with our covalent labeling data. And so we prefer this model, although this certainly is an alternate, but our covalent labeling data is not consistent with this model. And so we think we've begun to develop a model of what the dimer might look like. Now, of course, what we can do is test these models further, and we have done other experiments like that. So for example, in the side-by-side model, there are 2 pairs of salt bridges that are important for maintaining the interaction between the 2 monomers to form the dimer. And if you vary the ionic strength and do this reaction, you see that there's a strong dependency on the ionic strength for how fast the dimer forms, suggestive that these salt bridges are important. You can do mutation analysis to get rid of key interactions and so forth. And so we've developed some confidence that these models are pretty good of what the dimer looks like. We could do other experiments too, and I just say this is really a nod to some of the ion mobility work that Brandon described, is that we can also do ion mobility on this, where, as he mentioned, you can separate ions by according to their size and shape, and this can be particularly helpful for things that could conceivably be conformational isomers. And so, for example, if we take the dimer of Beta2 Microglobulin, do native spray, do ion mobility, we actually get a single peak, and if we can measure the collisional cross section, which is essentially a measure of the size, the rotationally average size of the protein dimer, it's more consistent with what you would calculate with the collisional cross section of this copper bound side by side, rather than this head-to-head interaction. Now, one thing I failed to mention, the other piece of data that, in addition to this and the covalent labeling, that gives us confidence in this model, is it turns out the copper is really important for this side-by-side model, and that it's actually not stable computationally without the copper. But in any event, these combination, we feel, of this covalent labeling plus modeling plus ion mobility gives us greater confidence in what this dimer model is. of course, we can go further and look at other oligomers. And so the tetramer ends up being a very interesting one, not only because it proceeds from the dimer, but it is probably, or at least we now have, show you in a moment, we have evidence that it exists in multiple different forms. And really interestingly, and this has been something that has intrigued us for a long time, is that copper is lost from the tetramer. We measure copper-free tetramer that is necessary before it can continue on the amyloid. So this transition is, we find to be very interesting. And so we've been lately very interested in trying to figure out what the structure of this, what this transition looks like, what are the structural features associated with the loss of copper for this to occur. And again, we're going to use covalent labeling and other tools that I just talked about. So let me just quickly tell you about the covalent labeling data. This is now a little bit more challenging to interpret because we are now looking at when the tetramer is formed. As soon as it starts forming in solution, we are going to be doing these labeling reactions, but there's always a background of dimer and monomer around. And so how you interpret the data has to be done a little bit more carefully. But long story short, what we find is if we take and map the places where there's changes occurring due to or decreases in labeling, if we map this onto our dimer model, we find that most of the decreases now when we populate with tetramer in solution are occurring on these edge strands of beta-2 microglobulin, suggestive that we're going this dimer, which is in a kind of side-by-side interaction, the tetramer then is forming with edge strands in some way. Now, if we do ion mobility, we find some very interesting results. We find actually evidence that there are more than one conformer of the tetramer, just like might be suggested from this model of the tetramer with copper going to tetramers without copper. And ion mobility is very valuable in sorting this out. And in fact, it's uniquely capable of doing this. There aren't many tools that can do this. And what we find is the tetramer forms 3 conformations. And if we do these experiments with and without the addition of EDTA, and so the EDTA is one of the key ways that we are able to populate this solely populate this sample with the copper-free form. What we see is that these 3 tetramer conformations shown in, well, gray here under normal conditions go down to 2 conformations when we add EDTA, suggesting that these 2 forms, what I'll call a more compact form because it has a collision cross section that's small, and then a more elongated form which has a larger collisional cross section. Those 2 forms remain, whereas the main form, the one that's populated most under the experiments, disappears. And so this is interesting. It says that the conformational change that occurs from going from a copper-bound tetramer to a copper-free tetramer forms at least 2 forms here, an elongated and a compact form. Now, I want you to remember this elongated form because it becomes very important later in the talk. So these presumably compact and extended forms are important for eventually forming fibrils. In what way, we don't know, but we're very interested, of course, in getting some structural insight into them. And we've begun with modeling and docking like what we did before. One of the things I failed to mention earlier with computational work is that we do the docking and the modeling in the presence of solvent because we're trying to model, of course, things that are going on in solution. But then when we compare them to, for example, ion mobility data, we remove that solvent, do additional molecular dynamics to get collisional cross sections that we can compare computational results to experimental ones. Now, what we have done is we've been able to develop a series of models for the tetramer. And right now we're trying to devise experiments to test each of these models because they can right now be best separated by ion mobility. We're starting to consider some of the tools that Brandon, for example, talked about in his talk, one of them particularly collision-induced unfolding, to tease out some other structural information. We're also starting to look at mutants that we mutate out certain residues in these models. And so we're hopeful that we can get some further insight into these 3 different types of conformations. So that's a little bit about what we've learned about the oligomers that precede the amyloids and some of the covalent labeling tools and ion mobility and molecular dynamics that we've been able to kind of provide some deeper insight into them. What I want to say now is a little bit about this kind of first step of the amyloid forming process for beta-2 microglobulin that forms copper. So what has been known based on some pioneering work from Sheena Radford's group and also Andrew Marinker's group is that the, if you will, amyloidogenic switch that occurs, this first conformational change that happens that starts the process off, involves a cis-trans isomerization of the histidine 31 proline 32 amide bond. In wild-type protein, this bond is in a cis conformation, and whether it's initiated by acid or copper, it converts to trans, and that is an important conformational transition that occurs, because that conformational transition leads to a repacking of the hydrophobic core of the protein and its exposure of some residues that ultimately help the dimer form. In particular, there's a phenylalanine 30, which of course is very close to this, the bond and the proline bond that's in question. It is normally buried upon the cis-trans isomerization. It becomes exposed, and taking its place as a tryptophan 60, which is normally fairly exposed, which is unique for a lot of tryptophans. It gets buried into that pocket that phenylalanine 30 vacates, and that's amongst a series of other conformational changes that occur to head towards dimer. Now, one question we were interested in asking is, can we use covalent labeling perhaps to measure this burial of tryptophan? And if we did so over time, could we get some rate of that burial occurring? And so what we turned to is a different reagent. You can see it here, dimethyl 2-hydroxy-5-nitrobenzyl sulfonate bromide, or HNSB, which is much easier to say. HNSB is a very valuable reagent because it only will react with exposed, solvent-exposed tryptophans. And so as they get buried, the reactivity decreases. And so it provides a nice convenient way, and it's a very fast reaction, to monitor what this tryptophan is doing. You might ask, certainly fluorescence could do the same thing, especially if a tryptophan is buried and then is exposed and becomes buried, the fluorescence should change. But it turns out there's another tryptophan in the protein that is already buried, and so its fluorescence is dominated by that. And so this is a convenient way to measure the tryptophan 60 burial. And so what we decided to try, and actually Nick Ruotolo, who was a graduate student at the time, who's I think in the audience today, first did these experiments. And it's a pretty straightforward experiment in which we can take beta-2 microglobulin and either in the apo form with nothing added or adding copper, we can measure, by doing little quick reactions, measure the extent to which the labeling of tryptophan 60 decreases over time as a proxy of it being buried, and presumably a proxy of that cis-trans isomerization that is occurring. And so just some control experiments, we can do it with the apo, we can add other metals like nickel and zinc, which are not known to cause the amyloid formation. There's no change in the tryptophan 60 labeling over time. But if you add copper, as soon as you add copper, you start to see a decrease consistent with tryptophan 60 being buried. We can also do a positive control of this, where we can actually take the mutant delta N6 beta-2M, which I alluded to at the very beginning of my talk. It turns out that that proline is in a trans position, and the tryptophan 60 is buried or partially buried in this mutant. And so its labeling is already less. And so as copper, after adding copper, we see tryptophan 60 being buried over time. And what this conveniently does is allow us to measure a rate of this burial, which we can do. And that rate probably doesn't have a whole lot of meaning to you. But the question is this meaningful in the context of the amyloid forming reaction? And so we can do a number of things to try to test that idea. And one of them is we can look at Thc fluorescence, which is an indicator of amyloid formation. It's a dye used for that purpose. It turns out for beta-2 microglobulin, when the dimer is formed, the Thc fluorescence will change. And so we actually have a convenient way to measure the rate of the dimer formation. And you can see that the rate of dimer formation from Thc fluorescence is slower than the rate of this conformational change, which is what you would expect. Conformational change happens first, and then dimer formation happens afterwards. And so we think this is a pretty decent indicator of this tryptophan 60 burial, and we like to argue that it is somewhat of a measure of a rate of the cis-trans isomerization process. Now, we can do this reaction as a function of temperature and do an Arrhenius analysis. This just gives you some examples of the measurements we've done. And so if we do this as a function of temperature, we can measure a barrier. And so with copper, we measure a barrier of about 15 kilojoules per mole. We can get entropic factors as well using the Eyring equation. I won't go into detail about that. And we also can do it under different amyloid forming conditions. So for example, pH 3.6, low pH condition that has been studied quite a bit. We find that the reaction is very fast at physiological temperatures, but drops off dramatically. And so this temperature, strong temperature dependency speaks to actually a higher activation energy, higher than the one with copper. Now, to put this in context, a typical cis-trans isomerization barrier for a free peptide is in the range of 60 to 100 kilojoules per mole. And so the copper catalyzes it, clearly this reaction. Acid does not. In fact, perhaps you could argue that it's a little bit higher than that, and it may actually not be the rate determining step that we're measuring. In fact, that's one thing I'll throw out there. This needs to further work to really shed light on that, but copper presumably catalyzes this. Acid, perhaps something other than the cis-trans isomerization, is the rate determining step. It still occurs, but perhaps something else is more defining the rate at which this happens, just because it's higher than the typical. Okay, well, so maybe I'll end with this because my time is running short. So some of you, if you're familiar with Beta2 Microglobulin, might ask, well, what about this mutant delta N6? Now, I mentioned this at the outset, that it can be used to convert the wild type in a pre-ion-like fashion to amyloids. And it's been known that delta N6 mutant has been known for about 30 plus years. It's been found in fibrils of DRA patients, but it's for a long time just thought to be the result of proteolytic fragment in which the first 6 residues just are cleaved off. I mentioned just a little while ago, it has a trans his31 proline 32 bond, and so the tryptophan was buried, like I mentioned. But one of the interesting things about it is it can spontaneously form amyloids at physiological pH. And it can induce the amyloid formation of the wild type. It's very, very slow at physiological pH. It's slightly acidic pH. It's a little bit faster. And so that's obviously great deal of interest, especially since it's found in the fibrils of patients, that this could be a causative factor. So we've been interested in measuring to see if we can measure the tryptophan 60 labeling as an indicator of the cis-trans isomerization. And so under conditions that are described here on the left, and I won't go through them all, but basically we can measure when we have a 3:1 molar ratio of the mutant delta N6 to wild type at pH 6.2. We can measure the drop in the tryptophan 60 labeling as an indicator, we think, of the cis-trans isomerization, and the rate is this. We do this at several different temperatures. We get a barrier that's in the ballpark of what we saw with acid, but it has entropic factors that are much more like what we saw with copper, suggestive of a tighter type of transition state. So this can be also done if we do a seeding experiment where we take the delta N6 protein itself, form amyloids of it, and then after about 9 days, take these amyloids, sonicate them a little bit to generate so-called seeds. And these seeds can then be immediately added with the wild type protein at pH 7.4. And as you might expect with a seeded reaction, the drop of the tryptophan, or the cis-trans isomerization, happens very quickly. And in fact, in a way that is a very low barrier, much like what we were seeing with copper. And so not only does this give us greater confidence in the method that we're doing, but we think now we have a way to start to look at, for example, mutations to either delta N6 or the wild type to understand which interactions might be important for setting this barrier. And that's something that we are starting to do now. And just to kind of put it in context here with the copper and with the low pH, we have with delta N6 soluble interacting with the wild type, we have a barrier that's much similar to the acid, but entropic factors that are also like the acid, but the seeding is much more like the copper. All right, so that's the context again. So I think I'm going to probably end there. I was going to say something about our inhibitor work, but I'm going to pass through that right now. And just to get to a summary of some of the things that I've said, hopefully I've shown you that covalent labeling, mass spectrometry, or protein footprinting that some refer to can be a useful way to study structures of protein. And I think it's particularly valuable for studying the interactions of proteins. And from it, for Beta2M, we get some idea that the dimer has a side-by-side structure when it's initiated by copper, and the tetramer forms with these edge strands, although the tetramer seems to have a good bit of conformational heterogeneity. And our labeling, modeling, and ion mobility are starting to provide some insight into that structure, into those structures. And then finally, I mentioned about the activation barriers that we're able to measure with this initial amyloid structural change for Beta2M, that we've been able to use this covalent labeling approach to get at some ideas about what those barriers are. So with that, let me end by acknowledging the people who actually done the work. I already mentioned Nick Ruotolo is one of the attendees here that started some of this work with measuring the cis-trans isomerization. Tyler Mercinko, who is also in the audience, it turns out, I believe, did a lot of the ion mobility work on the oligomers. And I believe I may have showed some data covalent labeling from Vanessa Mendoza as well. And I need to thank funding from the NIH, and both for a grant for this project, but also training grants that have funded students in the past. And then also collaborators, particularly John Han Chen, whose group here at UMass has really helped us get good at doing computational modeling. So with that, I will finish and be happy to take any questions that you have.
Okay. Thank you, Richard, for that great presentation. So we have what looks like a linked series of 2 questions on the Q&A from Eshas. He's asking, or she's asking, what is the pH where EDTA has been used? And I think the reason for this question is copper-2 can form different complexes with EDTA depending on pH.
Yeah, so that when we did these experiments, typically what we would do is we'd start the amyloid forming reaction. And then at different time points, we could add EDTA. And so those amyloid forming reactions were under pH 7.4 with all the physiological salts and temperature that we normally like. And so we would add EDTA. And I don't remember how long we would let it incubate in the solution, but it would have been at pH 7.4. And the idea was that, and what we found, just a little bit background of the experiment, we found that EDTA would, if it was added early, it would reconvert everything back to monomer. But if it was added late enough, you actually would convert everything back to monomer, but you would have some remaining tetramer that would be present and some hexamer. And after a certain time, adding EDTA no longer prevented the formation of the fibrils. And so, but it was always at pH 7.4.
Okay, so in the absence of more Q&A questions, maybe I can take prerogative and ask.
Looks like there's maybe one more.
Oh, is there one more?
I don't know, Benju, is that question for me?
That's for Brandon.
My question.
Yeah, that's Brandon. Yeah, okay, good. That was just, thank you.
So maybe I can ask you a couple questions. So if you have that tetramer distribution that you showed us by availability, and you track that distribution out for longer time periods, do each of those conformational forms decrease at the same rate, or do they all decrease at a similar rate? Or are some of them more linked to fibril formation and some of them less so?
Yeah, so we've done that a little bit, although the time course hasn't been done as as precisely as we would like. But there are changes over time. I guess that's the short answer. One of the interesting changes that occur is there's even more heterogeneity. And this is a question I think that maybe I've asked you before, given your expertise in mobility. We actually see a broadening of some of the mobility peaks over time as well. So there are changes, but there's also broadening that occurs, suggestive of heterogeneity that occurs.
Interesting. So when you do your chemical labeling experiments, like you showed us or for the dimer. When you sort of pick a time point within the aggregation timeline for the beta-2, how do you ensure that you're not labeling some mixture of oligomeric states? And if you were trying to go in to, for example, just label the tetramer to get some chemical labeling information, how might you approach that?
Yeah, so so I guess the quick answer is we're always measuring mixtures. So we would love to have the capability to isolate one of them and then react them. And we actually have tried some of those things by, for example, doing some size-based separation, collecting the fraction of a particular dimer and just reacting it. And that's a challenging experiment. So mostly what we've done is done labeling along the time course as the oligomers are forming. So for example, we might add copper and immediately doing a labeling reaction, and then do a labeling reaction every 15 minutes after that as dimer grows in. And then we have to track labeling as a function of time to really tell which ones are decreasing as the dimer increases in solution. That's typically how we've done the experiment.
Okay. Another question from the chat. Ashtush Tiwari asks, very nice talk. Just a thought on the nature of the interaction of the dimers, end-to-end, side-to-side, or interaction at an angle. Can this impact the overall hydrodynamic volume of the protein and impact its parent molecular mass?
So it won't, so by mass spectrometry, it won't have any impact on its apparent molecular mass. We are measuring it's a physical property that's unaffected by orientation. It's the physics of the fields working on the ions. And so it's not going to affect the mass in that way. We rely on the fact that its volume, if you will, or its cross-section will influence the ion mobility measurement. And that's part of what we take advantage of to be able to separate out these oligomers. Of course, in solution, the hydrodynamic volume will affect, for example, because I didn't talk about this, but we always do size exclusion chromatography alongside of any native mass spec experiments we do. And certainly shape affects hydrodynamic volume in a way that can affect its solution time in size exclusion. But we like the dual use of both size exclusion chromatography and mass spectrometry to sort those things out.
Question from Nick Barotto. Is tryptophan buried in the monomer model you use to generate the head-to-head dimer model?
Yeah, good question. So it is buried in that model because what we did is we put in copper, and part of the structural changes caused by copper, as I talked about, are tryptophan being buried, and so it is buried in that head-to-head model, as well as the side-by-side one.
Another question from chat. Rakez Kaed says, what conditions drive side-by-side versus head-to-head oligomers? Which one forms fibrils faster? Thanks.
Yeah, that's an interesting question. So I'll just say right now, the side-to-side or side-by-side oligomers that we see for the dimer with copper present seems to be the predominant form. Although when we get to these tetramers, the models that we have developed contain head-to-head types of interactions as well as side-to-side interactions. And so we don't think the head-to-head is present in the dimer form, but when a top tetramer does form, we do start to get some head-to-head interactions, perhaps as two dimers come together. Other ways to generate beta-2M amyloids have found evidence for head-to-head interactions. And so those, different initiating conditions, not copper. So it's hard to answer that question with copper because we only see side to side. There is not head to head, we don't think, in the dimer at least.
Maybe I'll take the opportunity to ask another quick question. So in the context of those tetramers, again, where you see those multiple structural forms, have you guys thought at all about trying to deploy some cyclic ion mobility to select out each one and do like a CIU experiment on it?
Yeah, we would love to do something like that. In fact, so what we're trying to do now, short of having access to a cyclic one, of course, we could send samples somewhere. We actually have recently installed a surface-induced dissociation device, an SID device, in our Synapt. And our eventual plan is to do conformer selected SID. And Vicky Wysocki's group has shown some really nice work that shows the SID behavior is very sensitive to the nature of the interface in the oligomers. Perhaps you remember that SID things can dissociate into dimers and other things other than just ejecting out a monomer. And so you can get some other information about the interfaces. And so that's what our current plan is.
Yeah, that'd be a cool experiment, definitely.
We got the device working. We just have to get it working well enough.
Thank you very much, Richard. Let's open up the session for both the speakers. Questions for both the speakers. Maybe I can read the question for Brandon. You can address the questions in the Q&A box.
Okay.
Ahmed, a technical question. How much does the ionization process and buffer affect the oligomerization of the protein?
Yeah, I mean, I think probably both Richard and I have experience with this to a certain extent. So the buffer, I think, can definitely influence the oligomerization of the protein. The ionization process, we try to make sure that that end of it doesn't influence the data we collect. I mentioned in one of my other answers to questions that we try to make sure that we keep concentrations low enough where you don't see like artifactual protein complexes being formed, oligomers being formed during ionization. And that makes it challenging to make measurements on more concentrated proteins if you want to try and probe how that level of protein might influence the oligomerization process. But it's not impossible, right? You can normalize your data for proteins that you think you understand in terms of how they are meant to be monomeric at certain sort of levels and what can be used to sort of like to control there. So I would say we try to make sure the ionization process does not influence things, but buffer definitely can influence oligomerization.
Yeah, and can I elaborate on that if that's okay? Or Rams, were you gonna follow up?
No, go ahead. Yeah.
Yeah, I was just gonna elaborate and emphasize the concentration feature of this. That low concentrations tend to avoid non-specific oligomerization. So when you're studying a protein like we are, that's what we're most interested in. We tend to work at low concentrations and use a type of electrospray called nanospray, which really facilitates, or I guess limits the possibility of having these non-specific interactions. And it's protein dependent. So some proteins, the concentration you need before you see these non-specific interactions can be lower than others. So it really is a protein dependent system to some extent.
So how low is the low concentration? I know it depends on the protein, but just give a number. Can you give a number?
I would say somewhere in the low micromolar is pretty safe, typically. Going into like the hundreds of micromolar is probably where you start to see some of these non-specific oligomer formation events.
Yeah, that's exactly what I would have said too. For beta-2 microglobulin, we're always spraying under conditions that are below 10 micromolar. So
connected to that, Brandon, you showed a kind of complex POPC and the synuclein. We know synuclein interacts with the anionic lipids, POPC being zotrionic. How much is the interaction, you think? How strong is the interaction? And how did it survive through that?
Yeah, so I think that's a good question. So, I mean, others in our field have actually directly observed peptides within nanodiscs without dissociating them from the disk. Mike Marty, for example, at Arizona has used some pretty fancy forms of orbitrap masterometry to do this sort of work. And you can definitely see that when you ionize these types of structures, they form as these intact nanodisk peptide or protein complexes. And then the goal then is to use some sort of collisional heating inside the instrument to eject the peptides and the proteins off the disk so you can measure them in more detail. In terms of knowing the level of strength of the interactions present, I don't have a good answer for that, but I think one of the future things that we want to do is definitely change the lipid composition in those discs and ask questions about how that's influencing the CIU measurements we take and how that's influencing the oligomeric states that we detect in the experiment. I think that'll be very key. I ultimately want to try and create a disc that better represents some of the biological membranes that the synuclein would encounter in neuronal cells. And that's, I think, the end state really of those experiments.
So in our hands, we see the phospeptide nanodisk, the belt, the phospeptide itself binding to or interacting with A beta, for example, also for IAPP. So do you see any interaction with synuclein that you work with?
No direct evidence of 4F interacting with synuclein, no.
Okay. So there's a question from Dheeraj Krishnan. There's a question. Thanks, Professor Ruotolo, for such a nice talk. I wonder if it is possible to quantify the protein biologics as well as its protein target complex in a tissue from protein biologic treated mice.
Yeah, so maybe it's not something that we're expert in, I would say, but people have started to look at a native masterometry from an ion source called DESI, which stands for desorption electrospray ionization. There, you're firing electrospray droplets at a tissue, and then from that tissue, you can capture, for example, a biologic or a biologic bound to an antigen, and then lift those off into the masterometer for analysis. That's very early stage results, as far as I know, in terms of using like a native mode type experiment for doing DESI like that. You could also, I think, probably probe that in a more sort of like classical quantitative, like bottom-up experiment if you wanted to quantify just a biologic within tissue. I mean, that would probably be the more standard way of doing it. But if you wanted to capture the sort of data that I was showing, the DESI experiment might be the most direct way of doing it. But it's definitely would be, not standards. It would be a lot of, I think, basic research to go into how to basically do that, and then be sure that the quantitative data you're getting was representative of what was in the tissue.
So there are several questions from Bin Zhu. He's in the panel. I'm going to ask him to directly ask you. Bin, go ahead.
Hi, great talks. I have a couple of questions for Brandon.
Okay.
So in your first one is related to the boundary between oligomers and like mature fiber or protofilaments. So in your case, I think maybe it's protein dependent, but in your case, how many what's the size you're detecting for like oligomers, and up to which size it will become so big it will precipitate out as like a protofilament or mature fiber?
Mm-hmm.
Do you see some certain like size, like a 15 mer, 20 mer, 27 mer, something like that?
Yeah, this would be a great question to get Richard's take on as well. I think it's definitely going to be protein dependent, and I think it's quite murky, like this boundary you described between, what is a oligomer versus what is a protofilament. I think that's an open question that I think a lot of folks in the field are still struggling with trying to define. I would say, for the systems that we study, like synuclein and A-beta, for A-beta, you can certainly see, if you really dig, you can see hexamer. There's been reports in literature as large as dodecamer being observed by mass spectrometry. I think typically in our experiments, we don't see much beyond tetramer. What if that's the real boundary between protofilament and like a oligomer? I don't know, but that's like the extent of the oligomerization that we could usually observe for A-beta. For synuclein, we can easily see dimers and trimers. Beyond that, I think is very challenging. And again, I don't know if that's really the boundary between what was be a protofilament and not. We're just sensitive to those samples for those oligomers that are soluble in solution, right? If it's part of some coupled state that's going to be aggregation prone, it's going to fall out of solution. I think it's really tough to observe those directly by mass spectrometry. And I'm sure Beta2M has got a different set of oligomers that you observe, right, Richard?
It does, yeah. And I think that, the issue of what's a protofilament and what's the end of the oligomers is sort of like, the issue of solubility and filter, is something soluble or not? Well, a lot of times it's decided by if it goes through a certain size filter or not.
Yeah.
And so I think the definition is hard to pin down. I will say by the mass spectrometry techniques that we have used, for example, for Beta2M, as oligomers get bigger and bigger and bigger, they're harder and harder to detect. And I think that's primarily an electrospray issue, but it could also just be a concentration issue. They just become now very low concentration. So we have a concentration sensitive detector, and so they're not easy to see. Whereas you might be able to see something of that size if you dried them and did TEM, or you certainly could see them if you did dynamic light scattering. I think it's probably difficult to define what's a protofilament begins and a oligomer ends.
Thank you both. The second question is related to the first one. So when you, for example, for mature fiber state, will you still be able to detect monomers and dimers? I'm wondering whether it's reversible or it's irreversible.
These aren't experiments that we do typically, but there have been reports in literature for people doing this sort of work. I think Andrew Marenker, for example, has done this. Carol Robinson, my old postdoc advisor, has done this, where you can see evidence of recycling of monomers and dimers off of fibrils using mass spectrometry. And those, I think, are pretty powerful experiments to do. Obviously, that would be system dependent in terms of which information you can get off of those experiments, but they're not experiments that we do typically. Richard, have you had experience with those?
You know, no, we typically don't do the experiments, but I will say we have done some experiments that kind of get to that when we make our seeds. So at the very end of my talk, I mentioned how we take delta N6, make amyloids, and then make seeds out of them by sonicating them and so forth. But when we take those seeds, we often filter them with a 0.45 micron filter. And in that solution, there's no free monomer around of the delta N6. So it's whatever we've done to make the seeds has also not liberated free delta N6.
So suggesting maybe more likely it's irreversible.
Yeah, well, in that case, probably it is irreversible, yeah.
The third question, that question about your CIU, the stability, the method you developed, CIU. So how does it corresponding to other methods like CD or NMR? You know, those are temperature-induced unfolding events. I mean, are they kind of, they can correspond?
Yeah, we've not tried to take that type of data and correlate it to CD or NMR data. We have tried to do it with calorimetry type data sets, which I think is probably maybe a better correlation to the information content we're getting. It's not perfect, right? I mean, CD might be actually a really good correlation to try and build there. We've not tried to do that. But for melt temperatures in solution, we do see some correlations between the two. In other words, they track in many cases in similar ways. Other cases, they don't. And in the cases that they don't, we can actually resolve differences that we know is structural and that are related to sort of functional forms of proteins or biologics in solution that you simply can't resolve in calorimetry because the experiment is too low resolution to be able to do so. And you're limited in calorimetry and the other methods that you mentioned to just looking at what's in solution, right? Which sounds sort of strange to say, but in Masterometry, you can parse a complicated mixture and just focus in on the stability of one effectively microstate within that mixture in a way you can't really do via these other methods. So it's really hard in many cases to build like true correlations between the methods, because in one, you have the ability to pick apart a mixture into individual pieces, and in most others you don't. So, but in cases where we can like do really apple to apple comparisons, we do see some correlations, yes.
Thank you. Can I ask one more quick question?
Go ahead, yeah.
This is like for in vivo exam, like a sample detection. For example, we have, some amyloid protein and we do like a drug treatment. Will you be, right now, the current status of the technology, can they detect the conjugation of the drug amyloid complex from like an in vivo experiment, from blood from like an animal?
If you took mass spectrometry overall, I would say, including like the bottom-up methods that Richard alluded to during his talk, I think those would be probably able to try and take on that challenge. The native work that I showed, I think, is probably not quite up to that level at this point. I would say in cases where I've seen people try to take on like samples that have had either limited purification steps between a cell and the instrument, or maybe trying to directly take, basically, the supernatant or some sort of fraction off of a cell. What they're able to measure is simply the most concentrated elements of those samples. Like you can take, for example, red blood cells, and you can detect hemoglobin relatively easily from those types of cells. But that's like a sort of almost a cheating sort of version of that experiment. But I would say for the bottom, if you're just looking for a conjugation event and a target, then yeah, I mean, I think there are plenty of mass spectrometry approaches that you could use to try and tackle that particular problem using some sort of purification strategy that goes directly online into mass spec.
Thank you.
So Richard, there's a question from David. I think you answered that. Could you expand a little more on that. So the
Sure.
question is side-by-side parallel or anti-parallel structure?
Yeah, sure, absolutely. In fact, I saw that question. I just responded. So I think the question was when I talked about this beta-2M dimer forming, there's a possibility of it being parallel, meaning the arrangement is the same direction, or anti-parallel, and we find that it's anti-parallel. All of our data points to that.
Great.
The model is only stable, and or at least the modeling data points to it the most strongly, because that form is the only stable one. The anti-parallel is stable, the parallel one is not stable, at least by molecular dynamics.
So the next question is from Beena Krishnan. You can unmute yourself and ask question directly, Beena.
Hi, thanks for the great talk. I have a question for Richard.
Yes.
So,
Hi, Beena.
your trip, hi.
How are you?
I am doing okay, thanks. I'm not sure I would remember a long time ago.
Yes, it was a long time ago, but
it
no.
was. So your last part of the trip accessibility data, I was thinking about the entropy for the acid mediated versus the carbon mediated, right?
Mhm.
One looks like the entropy favoring the